Much research has looked to better understand the underpinnings of childhood temperament and the interplay between genetic, neurobiological, and environmental factors in shaping their personality and behaviors. For example, Kagan et al. Investigated the neurobiological sensitivity underlying childhood shyness, identifying that children with lower thresholds for hypothalamic arousal are more likely to exhibit inhibited behaviors that will persist later in childhood. In another study done by Gortmaker et al., they extended the investigation of biological influences of shyness by exploring prenatal exposure to varying day lengths. Their findings suggest that gestation during months of limited daylight is associated with an increased risk of shyness, highlighting the influence of environmental factors in conjunction with biological predispositions in behavior development. Another example of this is seen in a study done by Mary Rothbart, who further touches on the integration between early temperamental characteristics and environmental experiences, and how this comes to influence personality development. Her findings suggest that both genetic and environmental factors are crucial in shaping temperament and its long-term outcomes. These studies together emphasize that while biological factors such as genetic predispositions and neurobiological sensitivity play a significant role, environmental influences, particularly during critical developmental periods, are also crucial in shaping behavior and its persistence later in life. But what processes exactly are influencing our personality and temperament prenatally? Hedges et al. Found that just one exposure to chronic maternal depression increases the risk of child psychopathology symptoms, including externalizing, internalizing, predicting lower communication skills, and leading to reduced social skills by the age of 5. Participants were drawn from an ongoing pregnancy cohort, and at 6 time points they assessed maternal anxiety and depression 3 times during gestation and 3 times postpartum. Mothers also completed child mental health, social skill, and communication skill questionnaires when their child was 5 years old. In a meta-analysis conducted by Kurzakowski and Leshout, they highlighted that exposure to prenatal adversity has been shown to have a significant impact on the development of the HPA axis, which regulates the release of cortisol following exposure to stress in order to increase the physiological resources devoted to dealing with that stressor. However, they noted that many studies have the limitation of poorly measured or unmeasured confounding variables, particularly parental personality traits and measures of specific postnatal environmental exposures, which is something to think about for my own study. In another study conducted by Rudd et al., women completed questionnaires in each trimester of pregnancy, an evaluation at birth, a detailed follow-up assessment when the children were 4, and another at 6 years old in order to gather reports of past exposure to stressful life events, women's exposure to stressful life events, and their perceptions of distress during pregnancy actually independently predicted children's behavior problems and adaptive functioning. This means that each has a separate and distinct impact on the child's behavioral problems and adaptive functioning. So, the collective literature has demonstrated how maternal stress and varying external factors such as daylight exposure largely impacts child's cognition and personality. This leads me to question if children born with increased neurobiological sensitivity are more at risk for inhibition. Specifically, I am interested in exploring if children with heightened neurobiological sensitivity, when exposed to early postnatal factors like parenting styles, may have an increased likelihood of developing social anxiety disorder. This research would provide specific insight at the effects of heightened neurobiological sensitivity in shaping social anxiety disorder in children, potentially informing future interventions and preventative strategies. I predict that if children are exposed to higher maternal stress during mid-gestation and who exhibit higher neurobiological sensitivity, which could manifest as elevated cortisol levels, for example, then they are more likely to develop social anxiety by the age of 8. My sample size would consist of a diverse sample of pregnant mothers and their children in a longitudinal cohort starting at mid-gestation, which would be the 4th to the 5th months of pregnancy. As Gortmaker et al. Mentioned, exposure to varying day lengths during mid-gestation had significant impacts on childhood shyness, suggesting this period is critical for setting developmental trajectories related to social behavior. The cohort will continue through age 8 and will consist of women from various socioeconomic backgrounds, and will also aim to include various ethnicities to address the limitations of many studies focusing on weird populations. The longitudinal cohort will allow us to see how prenatal conditions influence not only personality traits, but the stability of personality traits across time. Similar to Henges et al. And Rudd et al., the mothers will be assessed at various time points. We'll assess demographic information to ensure generalizability and any potential confounds. The mothers will then complete the same prenatal stress assessments as seen in Rudd et al. Study, where stressful life events during pregnancy was assessed by women's reports using a list of 14 events from the Center for Disease Control and Prevention, and subjective distress during pregnancy will be assessed via women's reports on their perceived distress during their pregnancy using an adapted version of the Goldenberg abbreviated scale. These stress assessments will be completed from the 4th to the 9th month of pregnancy, and additionally, maternal blood collected during the 3rd trimester will be analyzed for cortisol levels due to its significant association with distress-related hormone levels in offspring. The next assessment will be done when the child is 8 months old. This age was chosen due to Mary Ainsworth, a developmental psychologist known for her work in the development of the attachment theory, finding that anxiety towards strangers is a common reaction of Western children around 8 months old. At this stage, we'll only be assessing the child's neurobiological markers such as heart rate variability and cortisol levels. The next assessment will be at 12 months, where we will conduct the strain situation procedure to look at early social behaviors and attachment patterns. This procedure will allow us to see how the child acts with a stranger in absence of the mother, and can provide valuable insights into how early attachment relationships influence later social and emotional development, especially as attachment theory suggests that early attachment experiences shape a child's ability to manage stress and form social relationships, both of which are directly relevant to social anxiety. The next 3 sessions will be consistent with the procedures used in Kagan et al. Study when the child is 3 years old. 2 separate lab play sessions with an unfamiliar child of the same sex and age with both mothers present will be conducted, with indexes of inhibition being the long latencies to initiate play, speak, and interact with the unfamiliar child, as well as long periods of time beside the mother. During the session, mothers will be completing a report of their child's behavior and self-perception using the Behavior Assessment System for Children, which is a standardized measure of caregivers' ratings of children's behavior, self-esteem, and adjustment abilities in the home and the community. The next session at 5 years old, where children will be observed in 3 different unfamiliar situations, where inhibition will look like long latencies to play with an unfamiliar child, as well as time beside the mother, spatial isolation, and infrequent interaction, as well as reluctance to play with novel toys, which is suggestive of risk in an unfamiliar lab room. The last session will be conducted at 8 years old, which includes 2 assessments. The first will be an in-lab play session, which will involve 5 to 7 unfamiliar children of the same age and sex. 50 minutes will include structured gameplay, and 30 minutes will be free play. Inhibition will be infrequent spontaneous comments to other children and long periods of isolation from other children. The second assessment will be individual testing with an unfamiliar examiner, where the inhibition index will be latency to the 6 spontaneous comment to the examiner, and the total number of spontaneous comments over the whole testing session. All sessions will include cortisol collecting, where we'll obtain saliva samples before and after each activity, and mothers will complete perceptions of stress questionnaires in the previous month using Cohen's perceived stress scale and their depressive symptoms in the past 2 months using the patient health questionnaire, both of which were used in Rudd et al. Study. Importantly, the study will include a control group, which will include mothers who experience low levels of stress mid-gestation, which will be determined via the self-assessment reports that they completed during pregnancy. Given that Henges et al. And Rudd et al. Found stressful life events and their perceptions of distress during pregnancy independently predicted children's behavior problems and adaptive functioning by 4 years of age, and that exposure to prenatal adversity has been shown to have a significant impact on the development of the HPA axis, I predict that infants who are exposed to higher levels of maternal stress during mid-gestation will show increased neurobiological sensitivity and a higher prevalence of social anxiety symptoms. This is because elevated maternal stress has been linked to changes in fetal neurodevelopment, especially in areas related to stress regulation and social behavior, as highlighted earlier by Kurzakowski and Lachoute. Additionally, heightened neurobiological sensitivity, which is more likely to result from such stress, has been shown to increase the likelihood of shyness, as demonstrated by Kagan et al. And Gortmaker et al. Therefore, in my study, I would expect that children whose mothers experienced high levels of stress during mid-gestation will display higher cortisol level reactivity and more inhibited behaviors, such as social withdrawal during a stress-inducing task. If my results support this hypothesis, I would expect to find a significant difference in cortisol levels and social anxiety symptoms between the high-stress and low-stress groups. More specifically, if children in the high-stress group exhibit both higher cortisol levels and greater signs of social anxiety compared to the control group, this may suggest that maternal stress during mid-gestation plays a critical role in shaping neurobiological sensitivity and the development of social anxiety. To conclude, should maternal stress during mid-gestation indeed be linked to heightened neurobiological sensitivity and an increased risk of social anxiety in children, we have an opportunity to shape early intervention and prevention strategies in meaningful ways. For example, tailored interventions could address the specific needs of various demographic groups. For instance, lower income populations who often face heightened stress due to things like financial instability could benefit from targeted stress reduction programs that address such needs. This awareness could lead to the development of more personalized treatment approaches, ensuring that interventions are effective across diverse populations. By looking at the role of prenatal factors such as maternal stress in the development of social anxiety, this research has the potential to contribute to a more comprehensive model of social anxiety development. Ultimately, these findings underscore the opportunity to improve outcomes for at-risk populations and deepen our understanding of the complex interplay between prenatal factors and child development.